back into microfilaments. Okay, so Friday we got started on going through the ins and outs of what determines the dynamics of microfilament polymerization and depolymerization. So the main thing to have taken away from that outline of going through it is to just understand that at the very, very, very basic level, intrinsic to actin and microfilaments, there's a profound bias that's intrinsic to the molecule itself, actin, that's going to significantly favor polymerization at the plus end, okay? So there's a bias that's right there that the cell is going to start exploiting and use it in a very directed manner, as you're going to see today, in terms of talking about how are we going to generate the directed movement or migrations of cells in response to an extracellular signal, okay? So before we get to that, though, is needing to understand that microfilaments themselves, okay, they're flimsy, okay? There isn't a whole lot you can do with just individual microfilaments because, in essence, their structure is very much like a string, okay? So what we need to outline here are the ways that cells can actually take microfilaments and put them together into different structures, all right? And the simplest, as you might imagine, if you basically have a bunch of filaments or fibers, is to take them and bundle them together and make cables, all right? Pack them together. And that's going to give a lot more strength and a lot more structural integrity to the structures that you're going to form that are microfilament based. So these are just some of the ones that you can find in a variety of different cells, all right? So starting off at the top, the first thing you can utilize that are shown up here are what are called cross-linking proteins. And that is what they can do is that they can grab a microfilament on one side, a microfilament on the other, and literally chemically cross-link them or hold them together. It bundles them up. The only difference between the cross-linking proteins that you're going to use here is whether or not the protein itself is short, very small, like fibrin, or if it's a larger protein like alpha-actinin. And the only difference that that's going to do is to take microfilaments and either bundle them very, very tightly together, like you're going to find in the microvilli of the apical membrane of intestinal epithelial cells. So there, you can pack a ton of microfilaments together in a relatively small area, and that's obviously going to provide a lot of strength to that piece of the plasma membrane that's being protected. The other thing that you can do, though, with alpha-actinin is you can bundle them together but keep them further apart. And where you see that predominantly is in muscle cells. And the reason is that the microfilaments in a muscle cell, a striated muscle cell, have to be put together to give strength and integrity to the cell itself, but they have to be distanced far enough apart so that the individual myofilaments can slide past each other. You basically have to leave enough room for the motor protein myosin to slide back and forth between adjacent microfilaments, and that's going to allow the cell to contract or relax. You're solving the same problem. You're just doing it with a slightly different dimension or size of the cross-linking component, okay? Now, one that we're going to talk about down here in a moment is this protein spectrin, which is going to be used in the erythrocyte. And what spectrin is going to do is it's going to allow you to take the microfilaments and attach them to a cable-like meshwork that underlies the plasma membrane of the erythrocyte. And now what that's going to do is to provide a cell that can be compressed. So it provides strength, but more importantly, it provides compressibility or flexibility. Okay? As we keep coming down here, we can also get very, very large networks, meshes, so to speak, of microfilaments. And you'll see how those are going to be generated in a little bit when we're talking about promoting growth in response to an extracellular signal. And what we want to do is to send out a branching network of microfilaments that can literally push a very large area of the plasma membrane towards the source of an extracellular signal. And those are going to come into play during cell migration. And then finally, down here is the largest protein within the human genome. It's the largest gene encoding a protein, and that's dystrophin. It's the causative agent of muscular dystrophy. And it turns out what dystrophin has to do is it has two main domains. One domain attaches to the plasma membrane, and then the second domain attaches to microfilaments. And the reason that that protein plays a critical role in muscle contraction is that it anchors the microfilaments to the plasma membrane. So in essence, what happens is that when the microfilaments slide past one another and contract, they're anchored to the plasma membrane, so the entire cell contracts and relaxes. In the absence of dystrophin, and it is such a large protein that, again, like CFTR, it's very easy, or like the LDL receptor. The larger the gene and the more functional domains it has, the more it can be inactivated by mutations. And that's what happens in dystrophin that makes it such a prevalent genetic disease. And what happens is that in the absence of functional dystrophin, the microfilaments in the muscle cell will contract and relax and slide past each other, but because they're no longer anchored to the cell membrane, the cell itself remains relaxed. So the cell cannot generate any force for contraction. And so that basically caused the debilitating wasting part of muscular dystrophy, and the muscles just atrophy away simply because you can't generate any tectonic force within those cells, okay? So I want to take a look at these spectrum cables, though, and go back to the erythrocyte, okay? So again, if you take a look at the erythrocyte, it's a nonpolar cell. It's about the most apolar cell you're going to find that's out there. It has no specialized regions of the plasma membrane, unlike intestinal epithelial cells. So any one part of that membrane looks like any other part. They're a biconcave disc, and the problem that your erythrocytes have is that during their roughly 3.5 month lifespan of passing through the circulatory system, is that the cells have to literally squeeze through capillaries, one cell at a time, so they have to go through in single file, and the diameter of the capillary is smaller than the size of the erythrocyte. So the only way they can get through is you have to compress them. And then when they come out into a larger blood vessel, they can spring back to their normal shape. So that flexibility is built in by taking the microfilaments, and instead of having them come up like spokes on a wheel, like you're going to see how they contact a non-erythrocyte cell for migration, instead, you've got this mesh of microfilaments that kind of go throughout underneath the entire plasma membrane. But they're held together by this cable-like protein, spectrin. So in essence, you have these cables that if you take a look at the inside of the erythrocyte, underneath looking up towards it, you see these cables. And what this allows you to do is to diffuse or disseminate the force or the pressure that the cytoskeleton is putting on the plasma membrane. So instead of having single little points of contact between the microfilaments and the membrane, we're spreading it out around the circumference of the entire cell. So we have all these adapter proteins that are involved, and the details don't matter. Although two of the proteins, I just want to point them out because we already referred to them earlier on in the course. The first one up here is an integral membrane protein called band 3. Why was it called band 3? Here's another protein called band 4.1. Well, back in the early days, when they were taking erythrocyte plasma membranes and separating the proteins on an SDS polyacrylamide gel, they didn't know what the proteins were. So they just numbered them starting at the top with the largest protein to the smallest. Band 1, band 2, band 3, band 4, band 4.1, you know. So that's how they got their name. They have absolutely no functional significance at all. They're just telling you what their relative position was on an SDS gel. It took a lot of years after that to work out what they're doing. Well, m4a, as it turns out, is a bifunctional protein. It's an anion transporter, so it's the one that's involved in exchanging chloride and bicarbonate. So again, remember that most carbon dioxide is carried through the bloodstream as bicarbonate anions that are out in the bloodstream. And again, the enzyme carbonic anhydrase will convert carbon dioxide in the erythrocyte to bicarbonate anions that are then exported out of the cell. They go through the bloodstream. You get to your lungs, bicarb comes back in. It gets converted back to CO2. You take a deep breath, and it's gone. But this transporter also has an attachment site for the protein ankyrin, which is an adapter for spectrin. Then over here is the other protein, the very first integral membrane protein we talked about, and that's glycophorin. So the carboxy terminal region of glycophorin is also an attachment site. As you can see up here, for not so much ankyrin, but you can see that basically it binds spectrin and the microfilaments themselves. So we get this nice cable-like structure under the plasma membrane. Now, it turns out there's a lot of components here that are involved in forming this structure. So that means that you can, in fact, have deleterious mutations that can inactivate some of these components. And when that occurs, you get deformed erythrocytes. All right? And the components that are generally most frequently altered in these individuals that suffer from these relatively rare forms of anemia are mutations in spectrin, the cable itself, this little adapter protein up here, band 4.1, that links the microfilaments to spectrin together up at the plasma membrane, or ankyrin, that binds these separate spectrin cables together. And what happens is, in the absence of one or more of these components now, you lose the flexibility and you lose the compact structure of the erythrocyte. And you can actually take a look at them. And what you can see is that in some instances, instead of being these nice biconcave discs that you typically expect to see for erythrocytes, you see these cells here that are very distorted. They're elliptical. In fact, they almost resemble the sickle red blood cells that you find in sickle cell anemia. That's also due to a structural deformity, but that's due to the aggregation of hemoglobin in those cells. This particular structure arises because of deformity in one or more of those proteins. So some of these individuals, they're going to be anemic because these highly elliptical cells, again, they can't compress when they go through capillaries, and so they rupture. So in essence, you're going to be anemic. The other type of deformities that you can have, and it's very difficult to predict going in which of these proteins is most important for determining the structure. Meaning you can have mutations in ankyrin that can cause either elliptocytosis, or you can have mutations in ankyrin that cause spherocytosis. The phenomenon is the same. The morphology of the cells is different, but the outcome is the same. In the case of spherocytosis, you have very, very large spherical erythrocytes. Again, they can't pass through the capillaries because when they're compressed, they rupture, break, and because of that, you have a form of genetic anemia. Okay? So there are important components in providing this cell's architecture, but most importantly, this physical property of compressibility. All right? Okay, so now we're going to get to the fun part, and that is getting cells to move, migrate, in response to an extracellular signal. The extracellular signals that are most typically going to do this are either going to be growth factors that play an important role in wound healing, ultimately are going to trigger cell proliferation. So think of this migration as a stopgap measure. Okay? It's basically to literally fill in the gap until cells can divide and heal over the wound. The other is chemotaxis. And that is cell migration in response to a signal. And in this case, the most common signal that you find is a bacterial-generated protein called lipoprotein A. And that potentiates the proliferation and the movement of macrophages to sites of inflammation. Okay, so a couple of cells here that we can see. Here's a macrophage. There it goes. And the bottom one is a keratinocyte, or a skin cell, that's also migrating towards a growth factor. A couple of things that both of these illustrate is, can you guys see that you're pushing that leading edge across a very, very wide front? So you're talking about that's not having sort of a little finger-like projection of microfilaments on the plasma membrane, but you're doing it over a very broad surface area, which means that the microfilaments have to branch. So they not only have to grow, but they have to do so in a branch mechanism. Okay? So we'll see how these different signals are going to do this. The other thing that you can really see at the bottom with the migration of this keratinocyte or skin cell is, look how the trailing edge elongates. So as the leading edge is moving forward in this direction, the trailing edge, the cell is stretching, stretching, stretching. And it can't get too far ahead of itself. So there has to be crosstalk between what's happening at the leading edge of the cell, which is trying to move in that direction. But at some point, it has to cause the microfilaments back here to depolymerize or become fragmented and then detach, so that the trailing edge of the plasma membrane can keep up with the leading edge, and so the cell doesn't actually split itself in half. Okay? The key players that are involved in directing all of this migration are one of three small Rho-class G proteins. They're lipid-anchored proteins in the plasma membrane. Okay? They're entirely on the cytosolic leaflet. Now, in the signaling section, we'll get into how they're activated, how a growth factor or how a receptor is going to activate these G proteins to do their job. Okay, we'll get into that down the road. The activation of all three of these G proteins, Rho was the prototype, the first one discovered, so that's why they're all called Rho class. The other player is RAC, and the other one is a protein called CDC42 for cell division cycle gene. It was first discovered in yeast, and it plays a role in cell division that we'll get to also in a few weeks. Okay? Here's the key. Every one of these G proteins in their inactive state, like all the others, has GDP bound to them. So the binding of a signal to its receptor, first thing it does, it promotes the exchange of GDP for GTP. Been there, done that, there's nothing new. Okay, it's the GTP-bound form that's going to act to nucleate the formation of microfilaments at the leading edge of the cell, out here. The only G proteins that are going to be activated are the ones that are in physical proximity to the receptors that bind the growth factor. So these G proteins in this cell are all responding to the growth factor out here. The guys on this part of the cell do not. That's what provides the directionality. Okay? So think of the G proteins as distributed around randomly around the circumference of the cell. We're only going to activate and convert the ones out here to the GTP form. And what they are going to do when they're bound to GTP is that they bind one of three different adapter proteins. Rho, as you're going to see, binds to the adapter protein formin. Formin is going to nucleate the formation of linear microfilaments. So we're going to get a cell to stretch out and get longer. Okay? CDC42 and RAC interact with a pair of adapter proteins, WASP or WAVE. You can think of them as being functionally equivalent. So don't get hung up over that. They, in turn, are going to bind a pair of actin-related proteins, ARPs. We got ARP23. These are actin-related proteins, and what these guys are going to do is to promote the formation of branched microfilaments. Okay? Now, what you notice up here, and we're not going to get into any of the details in this unit, there's crosstalk among these three G proteins. And that makes sense, because what you're doing is you're having to coordinate the extent to which the cell is going to elongate, become skinny, at the same time that it's forming branched microfilaments and propagating the leading edge on a wider front. So there's crosstalk there. So these different G proteins are ensuring that the cell is not just pushing out on a very narrow front and elongating, but also that it's branching. That's part of the crosstalk. The other thing that these G proteins are doing is that they're making sure the leading edge doesn't get too far ahead of the trailing edge. So it's going to be having effects back there at the trailing edge that are going to entail the fragmentation and the depolymerization of microfilaments, and it's also going to affect the ability of the cell to remain attached to whatever solid substrate it's on. Okay? Don't worry about the crosstalk. We're not going to be getting into that. The thing that I really want you to take away from this little section that we're getting into is how do we get the directed movement at the leading edge? What is going on to get the microfilaments to polymerize where the G proteins are activated? And think about the consequences. How many ways can we screw up a G protein's function? You guys know that, come on. Two things you can do to a G protein that will perturb its function. What's the first thing you have to do to get a G protein to work? You have to exchange GDP for GTP. Okay, so start thinking. What will happen if that doesn't occur? If we have a mutation in a G protein that can't exchange GDP for GTP, guess what? The cell ain't going to move. Okay? The second one is going to be even more entertaining. And that is, what happens if a G protein can't hydrolyze GTP? Guess what? Now you're going to have G proteins lit up all around the cell, and you're going to have a cell that's trying to move in every direction at the same time. It's basically going to try and blow itself apart. And so either one of those is going to have consequences on the ability of a cell to migrate, but also on its morphology. So that's just, stay tuned, that's coming up in a minute. Okay, here's how we're going to make linear microfilaments. The first guy that's coming into play here is that little protein we talked about on Friday, and that's profilin. Remember that what profilin does, it accelerates the exchange of ADP on actin for ATP. So the first thing that profilin does is it generates a very, very robust actin that wants to go somewhere. That's the first thing that it does. It creates a pool of ATP actin that's ready to be used. But remember, profilin remains bound to the actin, and it generates a bias. So now what you're going to have is ATP actin that's bound to profilin can only be added at the plus end. You can't add it at the minus end. So now what we're going to do is take advantage of that. And that is, in response to activation of Rho, we've got this little adapter protein called formin. And formin is shaped like a paperclip. And it's normally folded over in its inactive form. The RBD stands for Rho binding domain. That is the region that can only interact with Rho when it is bound to GTP. So when you got the GDP form of Rho, it can't do anything. So formin is inactive under those conditions. So we get the GTP bound form, which means a receptor on this part of the plasma membrane was activated. When it binds to the Rho binding domain, formin elongates. It's literally like taking a paper clip and pulling it open. And now what it uncovers are FH1 and FH2 for formin homology regions. And all that means is it creates two binding sites that have a very, very high affinity for profilin actin. And what it's going to do is, let's say we've got a microfilament that's coming up here. So the plus end is coming up towards the plasma membrane. This FH2 region provides a high affinity binding site for ATP actin. So it recruits it. But now what it does is that the FH1 region is a loading site that loads up profilin actin. Remember, this guy can only bind to the plus end. Well, now what it's doing is that we're increasing the concentration of the plus end of a microfilament in very close proximity to the activated G protein. And what it does is that the profilin actin binds to FH1, and it's immediately shunted over here because it interacts with a very, very high affinity at the plus end of that microfilament. It's going to start growing. And what you see is what formin looks like. It forms basically a column. So it's a ring that's around the outside of the plus end. So you can constantly add more and more actin to it. Now look what's going to happen. Can you guys see how we're connecting the jigsaw puzzle? The microfilament is growing at this end. It's attached to FH2. It's attached to the RBD. So basically what we're doing is we're growing microfilaments here. It's pushing on this. It's pushing on that. This is anchored to the plasma membrane. So can you guys now see that mechanically, what you're going to do is that as this microfilament grows, you're now pushing on the plasma membrane. But you're just pushing it at one place. Because all we're doing is we're making a linear microfilament here. So now what we're doing is we're making long microfilaments, and they're pushing out on the plasma membrane at specific points. That's going to make the cell elongate. Okay? So that's pretty simple. And that's pretty much all you have to be concerned with what Rho is doing. What if we want to make branched ones? Now we're switching over to the other two little G proteins, CDC42 or RAC. Okay? What these guys are going to do, don't worry about this mechanism up here. All we're focusing at is that CDC42 or RAC are going to do the same thing to generate branched microfilaments. That's all we're doing that's differently. And the way we're going to do that is we have two different adapter proteins. The adapter proteins here are either WASP or WAVE. Doesn't matter. You guys can think of them as interchangeable for this discussion. So now we do the GTP form also binds, that's called the Rho binding domain, again, because it was first discovered in Rho. So they didn't come up with a new domain for every single one of these. It's a paper clip. It unfolds. And now what it does is look at the difference, though. This has this arm that swings out to the side and comes down and binds not at the very end of a microfilament, but a little ways down off to the side. And in fact, what it does is it's offset. If you take a look at the spacing there, it's offset by 70 degrees. It's not quite perpendicular. It's basically going to be at an angle. And what it does is that these adapters bind the actin-related proteins ARP2 and ARP3. If you take a look at them, they look just like actin. And in fact, they will do the same thing that actin does. Only now what you can do is ARP2 and ARP3 are nuclei that are offset at an angle. So what you're doing is you're literally seeding the growth of a brand new microfilament now. Only you're not making it from scratch at the very beginning. You're going to send a new microfilament off at an angle, 70 degrees. As this microfilament comes up here, it also can interact with another CDC42, another Rho binding domain, and in essence what we can do is repeat this process of branching at multiple points. So what Rho is doing is sending up a single microfilament that's going to push on the plasma membrane at one point. Can you see what these guys are doing now is that this is how we're getting branching. Because now what we can do is we take a microfilament that's going to come over here. Another one's going to come off it and push over there. Now what you're going to have is that these branched microfilaments themselves run straight into the plasma membrane. So they are physically pushing on the plasma membrane itself, only now they're doing it at angles to where the original microfilament came from. This is what's going to allow the cell to now migrate across a very, very large surface area on the plasma membrane. So we get it crawling, we're elongating the front at which the cell is going to migrate forward. The two processes are going on at the same time. Okay? So, what we're doing then is that all of this is happening down here at the plus end of the microfilaments. So we're getting these little pseudopod or philopod projections first, where it's kind of like the cell's putting out these little individual runners. It's kind of creeping forward. We get these long stress fibers. Those are all generated largely through the action of Rho. The cell is stretching out. Then CDC42 and RAC are causing the cell to branch out and move. So it's a coordinated motion where the cell just starts sending out little processes, elongates, and then it gets wider. The extent to which this happens in different cells, that's intrinsic to the relative numbers of the three different G proteins. Some cells will get longer than others because they have more Rho. Other cells, like that macrophage, will branch out more because they've got more RAC or CDC42. You just control the morphology of the cell based on the numbers of the G proteins that are being activated. Now, the only thing to talk about back here at the trailing edge, and we're not going to get into a lot of detail other than what's shown here, is that cells normally have integral membrane proteins that come out and they're called integrins. And they're adhesive molecules. The integrins bind to proteins that are underneath the cell surface. Usually it's the protein fibronectin or the protein collagen. And it allows the cell to kind of glue itself down and fix itself at one place. These proteins are often perturbed in cancer, and it makes the cell slippery and more readily detached. What happens as part of this concerted movement, so we've got all this microfilament growth and branching that's taking place up at the plus end. What's going on back here at the back end is that these G proteins are communicating a signal to the motor protein myosin that's literally causing the microfilaments in the back of the cell to slide past one another and contract, just like they do in a muscle cell. So your cells that are migrating are contracting just like a little muscle. But what has to happen is that the integrin proteins are also being phosphorylated. And when they're phosphorylated, they detach and let go of the underlying substrate, so the back half of the cell picks itself up off of its surface and then rushes forward and catches up with the leading edge. And that's what keeps the cell from ripping itself in half. Okay? That's pretty much all that's going on. Now, here's how you can mess it up. You can go into cells and you can knock out the GDP exchange activity of any of these G proteins. You could take out the GEF, or you can inactivate the ability of any of these G proteins to exchange GDP for GTP. You now take these mutant G proteins and put them into a cell. And the assay that you're using is to artificially mimic a wound. So what you do is the following. If you take normal cells and grow them in culture, they grow to what's called confluency. Tumor cells don't do this. If you're going to see tumor cells lack the ability to form a confluent monolayer, and instead the cells pile up on each other and form little tiny tumor-like structures that are called foci. Okay? But normal cells grow to confluency. And that is every single cell here is surrounded on all of its sides by another neighbor. It's literally a tissue that's one cell layer thick. That's what normal cells do. If you take a confluent monolayer of cells in a petri dish and take a spatula, a rubber spatula, and come through and scrape away a line of cells, you just mimic a wound. It does the exact same thing that would happen if you cut yourself. You've now got a gap where the cells are no longer confluent. And what's going to happen is the cells that are adjacent to the gap perceive the absence of neighboring cells and they start secreting growth factors. Okay? Ultimately, those cells are going to divide so they can fill in the wound. But in the short run, the growth factor causes the cells to move in and migrate and help fill in the wound. So what you can see is normally this process, here's a gap that you could create. You're looking at the cells here and here. And what you can see is within about 3 hours after the wounding, the cells have already started migrating. It takes about 24 hours before those cells start dividing. So right here, they're just migrating. So here's what you can notice. If you take cells and you're doing this little simple wound healing assay, and the cells are now expressing what are called dominant negative forms of the G protein. What do you think the negative means? Let's go through this. We have to spend a little bit of genetic terminology here. Why would these be called negative? They lack the ability to exchange GDP for GTP. So why would we call that negative? They don't work. Okay? So it means that that type of mutation of a G protein doesn't work. It lacks any activity. Now, why is it called dominant? When we go to genetic speak, and we use terms like dominant and recessive, what are we referring to? Remember that what you're talking about is dominant and then recessive refers to the phenotype you observe in a cell that's expressing both the mutant allele and the wild type allele. So in these cells, think of it this way, they're heterozygous now. They've got a wild type allele of a G protein that can do its thing, but they also have one of these mutant alleles, okay? They cannot exchange GDP for GTP. And we see a dominant negative phenotype in that case, meaning the cell behaves as only as if the mutant is there. And the reason is there are not enough of the wild type G proteins to cover for you. Okay, so you don't have enough of them there. So in this case, this is why these are called dominant negative. Dominant because you see the phenotype of the non-functional form of the G protein. So if you take a look here, and what you're measuring is in control cells, over a period of time, you'll get complete wound closure. But if you take a look, if you have these dominant negative forms of any one of these individual G proteins, you don't get the wound closure. Now, can you conclude anything here about which of those G proteins is most important for wound healing? If you take a look at the graph, which one seems to have the predominant role? Sorry, Freudian slip. Whoa. So getting those linear microfilaments started plays a critical role in getting cells to migrate. RAC and CDC42 are less important, but it's also because they're redundant with each other. These two G proteins can carry out about the same function. There is no backup for linear microfilaments other than Rho. Okay? So this is what happens if the G proteins can't work. What happens if they try to work all the time? So now what we're going to do is introduce into cells mutants of each of these G proteins where instead of not being able to exchange GDP for GTP, they can't hydrolyze it. And now that's what you see. Okay? These are the microfilaments that you form in these cells when you cannot hydrolyze GTP. Here's the control cell. So that's a normal, healthy-looking fibroblast where you've got normal G protein function. Look what happens if you have a dominant active Rho. Now you notice it's not dominant negative. It's dominant active. And what that means is that the mutant allele is always on. It's always got GTP bound to it. That means the cell can no longer respond in a directed manner to an extracellular growth factor because it's making linear microfilaments all of the time, everywhere. The cell literally fills up with all of these linear microfilaments. If you take a look at RAC or CDC42, now what you're doing, and you can really see this with CDC42, you're forming branched microfilaments now that are pushing out on the plasma membrane in every direction. It would be as if that cell is surrounded by an extracellular growth factor instead of it just being produced on one side of the cell. So in this case, for both of these cells, what it's trying to do is the plasma membrane is attempting to try and reach out and spread in every direction simultaneously. You have to control how you express these proteins in this cell, because if you leave them to that long enough, the cell will literally just rip itself apart. Okay, the plasma membrane will literally try and go out in every direction. This one will try and elongate in both directions, and eventually the cells will die because they literally fragment their microfilament, the plasma membrane, from trying to elongate in every direction. Okay? So, you can see there's a very, very profound difference between a G protein that can't exchange from a G protein that can't hydrolyze. Okay? All right. To finish up, let's have some fun. So here's a competition. So a bunch of scientists several years ago, back during the last Summer Olympics, the London Games in 2012, they decided to organize and put together an international competition. And what they did is they laid up some very specific ground rules. So what they did is that everybody could take their favorite cells that they were working on in their lab, put them into Petri dishes that were coated with a very similar substrate, so there's no performance-enhancing drugs, there's no growth factors, no stimulants, it's just fibronectin. And what they're doing is that they wanted to determine, of all the different cell lines that are out there that people study, which is the fastest in the world. Okay? They wanted to see who's the Bolt, who's the sprinter. And so they actually put this through kind of a joking type analysis with a lot of different laboratories, but they actually compiled all the data and put it together and made a video of this. And so, just to show you, here's what the races look like. Okay, so we're going to start with the preliminary heat. All right, so here's the preliminary heats. So we got a lot of competitors from different nations. Turn it off here. And the first thing to notice is, if you take a look over here, these are the names of the cells. Okay, and you can see different sources. So we've got, E typically are embryonic cells. You notice a lot of these with the ABC. Those are tumor cells, adenocarcinoma cell lines. Fibroblasts, they're all color-coded. Now, the one thing you guys notice, these are intrinsic motility assays. So these are cells that have the ability to migrate on their own, even without being exposed to an extracellular growth factor. So it's not surprising that most of these cells are tumorigenic. They're derived from cancers. They've been transformed. Because one of the features of metastatic cancer cells is they have the capability of migrating on their own. But you notice some of them are a little confused. You notice how they kind of go back and forth. That's because there's no directionality to it. It's basically whichever way the microfilaments start polymerizing one side over the other, that determines the way that they're going to go. Okay, so these were the preliminary heats. Okay? So now they're going to go through, you take your top 4 finishers from each group, you put them into the track, and now we're going to look at the final. Okay, here we go. Here's the finals. there's our winner. Not even close. And as it turns out, if you take a look, it's a primary embryonic cell line. So these are naturally migrating cells in developing embryos that have the intrinsic ability to migrate really quick. That's the bolt. That's the sprinter right there. The other thing you notice how those cells, you see that birefringence, you notice that you see that refractive rounding up. That's because when these cells migrate, they round up and become very compact. That is also going to be a property that you're going to see of cells that undergo mitosis. It's also going to be a recurring property of metastatic cancer cells. Okay? All right. That's a good place to finish for today. So on Wednesday, we're going to start in with microtubules and looking at motor proteins there, okay?